Hello, we are Longevity Genie team, and we want to present our Cell2Sentence4Longevity project. So what we did, we took C2S scale Gemma to 27 billion model that could do both general LLM inference and single cell RNA-seq analysis. We fine-tuned it with age-labeled scRNA-seq data that allowed us to make it useful for aging research applications like identifying age-associated cell changes and perturbation predictions. So what is single-cell RNA sequencing is when we take a tissue, we split it into individual cells, we take those cells and we sequence it, and we know expressions of genes on individual cell level that can allow us reasoning on cell subpopulations and individual cells. The approach to fine-tune general purpose LLM is to turn gene expressions of each cell into sentences when we rank genes by expression and concatenate their gene symbols. Why it is useful? Because general purpose LLM already can see experimental descriptions, donor health records, and we extend it with single cell RNA-seq understanding capabilities, and with our age prediction fine-tuning. It now can also say which cells age faster. It can simulate perturbations like adding drugs, and it can characterize aging at cell population level. So for this project, we wrote a whole data processing pipeline that does conversion of single cell RNA-seq datasets to cell sentences. It makes balanced splits and uploads everything to Hugging. We also release datasets large for LoRa fine-tuning and small for full fine-tuning that we eventually used, as it yielded better results but needed more compute. So our fine-tuned model, it can actually predict age better than state-of-the-art in terms of mean average error and root mean square error. However, the Pearson coefficient still needs improvement, and we believe that improvement can be reached because of full fine-tuning. We only used very small subset of all available data, and our training loss didn't really reach the plateau. With more compute, we can do better. And the most useful out of it is that actually results of our model have biological meaning. So what we did is so-called in silico knockout analysis, when we compared predictions with and without some highly expressed genes. And the genes that change age prediction most of all, actually they aligned with known biological literature. That means that probably we can suggest some experiments to extend lifespan. We also looked on drug perturbation, how cell state will change if we give drugs to those cells. And obviously we wanted to see how we can develop interventions based on our insights. So we wrote a whole longevity forest agentic pipeline that could run our model to get in silico knockouts. Then it used the pipeline to analyze and annotate genes with multiple databases, and at the end it used Protein Hunter MCP that we also developed to actually develop peptides with degradation tags to destroy unwanted proteins. We provide the video demo of the pipeline, and also Protein Hunter is released with MCP. That means that you can run the analysis in your cloud desktop or cursor, for example. So here is our team, and thank you very much for your attention.